Howdy, this is Lab Report 13, PCR and gel electrophoresis. This is Group 2, Diego Castillo, and myself, Rita Cantu Mahurin. In the introduction, we highlighted the importance of this lab, which was the uses of molecular biology techniques to identify and analyze DNA using three major steps, which are PCR, and this amplifies a specific region of DNA, the restriction digestion, which cuts DNA only if a specific sequence is present, gel electrophoresis, which separates DNA fragments to reveal whether this DNA is detected, and the overall goal of the lab was to use these DNA testing techniques to determine the genetic identity of the sample. The materials we used were the DNA samples, the PCR reagents, which were the master mix, primer mix, the PCR tubes. We also had a restriction enzyme, the mini PCR thermal cycler, the electrophoresis chamber, the TBE buffer, the agarose powder, the cyber green dye, the fast DNA ladder, the micropipettes, and the sterile tips, the heat-safe flask, the microwave, and of course our PPE. In the procedure, this was a continuation of what we did in the previous lab section. So we had originally utilized this restriction digestion, which was labeling the four PCR tubes R1 through R4. And then we used a new pipette tip for each sample, transferring 15 microliters of PCR product from the tubes 1 through 4 into their matching labeled tubes. And then we added the 2 microliters of the diluted restriction enzyme to each tube. We closed the cap securely and flicked the tubes to mix the reaction, and then we wanted to also ensure that all of the liquid was at the bottom by also briefly spinning the tubes if necessary. And then we did incubate them overnight using a mini PCR and heat block mode, a water bath, or an incubator. And then following this was the next lab time, which was when we prepared the gel. So we added 50 milliliters of the TBE buffer and then 0.75 grams of the agarose powder to the flask, and we swirled it. And then we also had the microwave mixture, which we only microwaved until it was just boiling. And then we waited for the flask to cool and we placed the DNA ladder into the second ring of the electrophoresis table. This is a little bit different for us since we were sharing our gel electrophoresis ladder with the other group in our lab group, so this will look different in our results. But once the flask was warm, we did add 5 microliters of the cyber green, and then we poured the mix into the table. And it was very important that we didn't touch the gel until it was completely solidified. And then as far as the PCR procedure, as I said before, of course, our wells were loaded a bit differently since we were sharing it with another group, but we did position the gel so that the wells were closest to the negative portion. We loaded 10 microliters of the DNA ladder, and then we loaded the rest of the restriction enzyme wells. We ran the gel at 100 V for 30 minutes or until the bands were separated, and then we were able to view it under the UV light.
These are the results for our gel electrophoresis. Our results are the left side of the gel. So 1, 2, 3, and 4 there, and 5, which is the DNA ladder. And then on the right side, that's group 1. So our focus right now is on the left side, which is group 2's, our group. You can see that the controlled non-pathogenic E. coli shows only one singular band around 400 BP, which matches what's expected from the control. As for the pathogenic E. coli, you can see that it does have two bands, or even looks like three bands, but it should have been two bands at like around 250 and 150 BP, which are lower than the 400. So yeah, that did work. You can see that they are lower than the non-pathogenic E. coli control. And now for the samples, the third column is the lettuce sample, and the fourth column is the tomato sample. You can see that the lettuce sample, which is the third one, did not have that E. coli because it only has that one singular line at 400 BP, while the tomato did have that sample, that two bands, like at 250 and 150, meaning that the tomato is contaminated with that pathogenic E. coli. We used the DNA ladder to compare both results. So basically, it just means that in the pathogenic E. coli, you can see that those two bands specifically, and you can see it in the tomato sample, meaning that the tomatoes are contaminated. So what would we do to prevent illness? Well, they would issue a recall of those tomatoes from Red Ripe Ranch, and notify those food trucks and the public, and they would try to trace contamination at the farm. So like irrigation water, fertilizer handling. DNA testing is used in health for detecting genetic diseases like cystic fibrosis or sickle cell anemia, identifying viral infections like COVID-19 PCR tests, even forensic analysis and crime scene investigation, and even screening for cancer mutations like BRCA1 or 2 for breast cancer risk.
In conclusion, this lab utilized PCR, the restriction digestion, and gel electrophoresis to determine the identity of a DNA sequence. The PCR successfully amplified the target region, and the restriction enzyme cut the DNA only if the site was present. And so the resulting band pattern on the agarose gel revealed how molecular techniques can, in fact, accurately identify genetic differences in the DNA samples. And it also demonstrated the power of molecular diagnostics and reinforced core skills in DNA handling, amplification, and electrophoretic analysis. Thank you so much. Once again, this is Group 2, Diego Castillo and Rita Cantu Mahurin.